Hello friends, in this session we will talking about the structure of DNA and RNA. So let's start the DNA first. So DNA is composed of polynucleotide chain. So we will simply define the structure of DNA before going to discuss about the definition of DNA. So if we talk about the structure of DNA, we have some basic points, and these three basic points are: first point is the sugar, second point is the phosphate, and third point is the sugar, phosphate and nitrogenous base. So these three things are required to make a DNA, and apart from them there will be the another things which will be required to make the DNA, and some enzymes, proteins are required to make the DNA. But if we focus the structure of DNA, then these three things are needed as the structure of DNA. So these three things which will be required. So first we will have to characterize these features. This will be the sugar. So sugar molecule are required to make a DNA and RNA. In case of RNA, there will be the ribose sugar, and in case of DNA, deoxyribose sugar. So ribose and deoxyribose sugar are required in DNA and RNA respectively. The sugar molecule is hydrophilic in nature. If we talk about phosphate, which will be the, or you can say phosphoric acid, which will also the hydrophilic in nature and present in both of them, DNA and RNA. And third, nitrogenous bases, or you can simply call it as a basis, because in DNA and in RNA there will be must presence the bases. In case of DNA, there will be presence of 4 bases: adenine, thymine, guanine and cytosine. In case of RNA, instead of thymine, there will be the presence of uracil. Rest of bases are as it as DNA. So these three things are needed to make the backbone. Sugar and phosphate are needed as the backbone of DNA, So this is the backbone of DNA. So if we want to zoom this portion only, so suppose we have the sugar and we have the phosphate. We have the sugar and we have the phosphate. So where the sugar present, this is acting as the 3 prime, and where the phosphate group is present, this will be acting as the 5 prime phosphate group. So 3 prime group is acting as the polymerizing end in the DNA as well as in the RNA. So we have this backbone and this, there will be the two polynucleotide chain wrap around to make the DNA, and two polynucleotide chain make the DNA, and there will be the presence of hydrogen bonding between the nitrogenous bases, which will make the complementarity to each other to form the coil-like structure, or you can say rope-like structure. In this manner, the DNA is a polymer of polynucleotide chain, and these two polynucleotide chains are needed to form the DNA. DNA is a double-stranded form of genetic material. So DNA can acting as a genetic material. Why we call it as a genetic material? Because of a DNA is a self-replicating molecule, or you can say material, that is present in nearly all living organisms as the main constituent of chromosome. It is the carrier of genetic informations, and some viruses also possess the DNA as a genetic material. So if we want to define the DNA, we have to define the DNA it as a deoxyribonucleic acid is a polymer composed of two polynucleotide chain that coil around to each other to form a double helix carrying genetic instructions for the development, function, growth and reproductions of all organisms and many viruses. So we have to simply call that some viruses have the DNA as a genetic material. So if we talk about polynucleotide chain and the structure of DNA, so the DNA, this is the backbone, will be the one backbone and this will be the another backbone. So sugar and phosphate make the backbone, and nitrogenous bases which will make or help in coiling around to each other with the help of hydrogen bond. So some important points which will be required. So adenine can pair with the thymine with the help of two hydrogen bond, and guanine can pair with the cytosine with the help of 3 hydrogen bond. If we talk about hydrogen bond, so there will be the one thing which will be required to make the hydrogen bond. First thing is the donor group, and second thing is the acceptor group. So one polynucleotide chain and another polynucleotide chain, one have the donor group and one have the acceptor group, to make them the hydrogen bond, to make them the stability to the DNA. And there will be the sugar phosphate backbone, and there will be the sugar phosphate backbone. These two sugar phosphate backbone and hydrogen bond to make them DNA to coil each other, and 3' OH end can only acting as the polymerizing end. 5' phosphate not acting as a polymerizing end. So this is the important, and DNA is self-replicating molecule, and this will be the genetic information. Suppose we have the DNA and we have to transfer the genetic information into the next generation. What we have to do? We have to do that first of all, this DNA have to be replicate first and then transcribe and then make a protein So the replicated DNA molecule gets transferred to its own progeny with the help of reproduction. There will be multiple type of reproduction mode, bisexual as well as an asexual mode. Both of them possesses DNA as a genetic material. So if we talk about structure of nucleotide, what to do to make a polynucleotide chain? We have the sugar molecule, we have a phosphoric acid, we have the nitrogenous bases. So these three things make the nucleotide. And if we want to remove the phosphoric acid or phosphate group, we call it is a simply nucleoside. So if we talk about the complementarity of two polynucleotide, so we have the two polynucleotide. There will be presence of some sugar molecule, some phosphate group, some nitrogenous bases. So if we want to form the double strand DNA, what we have to do? We have to do first of them, we have to form the adenine. We have to form the hydrogen bond. Adenine can pair with thymine and vice versa. Similar manner, guanine can form with the cytosine with the help of 3 hydrogen bond. And if we want to, who is the person which define the orientation of complementarity of DNA? There will be the presence of 3' OH, and where the 3' OH present? The 3' OH is present in the sugar. So this is the 3' OH which will define the polymerization or orientation of both the complementary polynucleotide chain. So if you have to see, this is the sugar, and in this strand the sugar is like this pattern, and this strand is sugar like this pattern. So 5' to 3' is acting as a leading strand. When we focus on the polymerization or application, we will learn about detail in the leading or lagging strand. So this is the 3' OH. This is the 3' OH. So 3' OH is acting as the extent strand or polymerizing end of the DNA. In similar manner, this strand is acting as the, but in this case this will happen this type and this will happen this type. So we call it as a complementarity in both of them, complementarity. So one strand should this type and one is this, not this possible, or not you have to this possible. This time you have to wrong, and this time you have to write them. So some chemical groups which will define the major and minor groups from the edges of the base pair. These chemical groups are present which will define the major and minor groups. So double helix exists in the multiple conformations. DNA is a double helical molecule, and this double helical molecule not form only in the B form of DNA. It will also form the A form. This will also form the Z form of DNA, and which will be the conditions which will define the A, B and Z form of DNA. So there will be the comparison structural properties of A, B and Z form of DNA, which will be derived from the X-ray analysis. So if we want to set that B form, we have to compare B form of DNA with both of them right-handed helix, which will present in the B form of DNA, and A form of DNA. Both of them possesses right-handed double helix, and left-handed DNA possesses the Z form of DNA, rise per base along axis. So this is the chart which will require to make or to solve the questions. Questions are coming from this part only. And glycosidic bond. Remember, where is the glycosidic bond? Sugar and nitrogenous bases are bonded or joined to form the glycosidic bond. So anti-glycosidic bond are formed in A and B form of DNA, and anti and C, anti and syn, both of glycosidic form in the Z form of DNA. If we talk about major and minor groups of DNA, so if we want to B form of DNA first, so wide and deep, its major group is wide and deep. So, and if we talk about A form of DNA, its major group is narrow and deep. And left-handed DNA have the flat major groove. So this chart will be more applicable all the exams and all the needed questions to solve with the help of this chart only. If we focus on the structure of RNA, polymerizing process, both of them same in the RNA and DNA, but in RNA there will be the coiling to polynucleotide chain, and RNA is a single stranded molecule and DNA is double stranded. RNA have multiple structures and diversity, but DNA is less than diversity and structure. RNA is unstable and DNA is very stable molecule.